Hello students, today I have come up with this video on class 12th Biotechnology, Unit 5, Protein and Gene Manipulation, and the chapter which I am going to deal is Recombinant DNA Technology. So again, this is a very interesting topic of biotechnology, so let's start. Let's have an overview of the chapter. So what we are going to learn in this chapter, number 1, we are going to have an insight into recombinant DNA technology. Number 2, we are getting an idea about what molecular seizures or restriction enzymes are. Number 3, types of vehicles for cloning, also known as vectors. Number 4, the basic steps involved in cloning. Number 5, the creation of DNA libraries. And number 6, the techniques like PCR, southern hybridization, DNA sequencing and site-directed mutagenesis. So students, everyday newspapers tell you about remarkable feats of science and technology in helping man combat disease and improve his environment. Many of these use recombinant DNA technology. Using this technique in the past, bacteria were engineered to produce insulin. You can see in the picture, the first picture on the left hand side. You can see the 1982 GMO insulin. It is the insulin which is human insulin which is produced in bacteria. Hepatitis B vaccine was again developed by the help of recombinant DNA technologies, and the cells which were used for producing this hepatitis B vaccines were yeast cells. Then you can see the cotton plant which is insect resistant, is also known as Bt cotton. Bt stands for Bacillus thuringiensis, the bacteria whose gene has been inserted into the cotton and made it insect resistant by producing a certain kind of protein which we will learn about in the upcoming chapters. As you all know that all the organisms have DNA as their molecule of heredity. And if we see it at a chemical level, DNA is the same whether it is taken from a microscopic bacterium or a blue whale. That means DNA is made up of the same chemical components, sugar, phosphate and combination of nitrogenous bases. As a result, DNA from different organisms can be cut and pasted together, resulting in recombinant DNA. This has been made possible by the recombinant DNA technology, or also called genetic engineering. This field of biotechnology encompasses a number of methodologies that enable new combinations of genetic material to be artificially constructed in the laboratory. So basically, with the help of recombinant DNA technology, we can combine the DNA from two different organisms into one, and that DNA is known as the recombinant DNA. Now we are going to have a look on what basic steps have been involved in this technology. So the first step is to isolate a DNA fragment containing the gene of interest that needs to be cloned, also known as insert. So if you want to clone a gene from a plant cell, so you have to take the DNA fragment from the plant cells, and if you want to clone the gene of interest from animal cells, then you have to take the DNA fragment from animal cells likewise. Number 2, generation of a recombinant DNA molecule by insertion of the DNA fragment into a carrier DNA molecule called vector. So what is a vector? Vector can be any DNA fragment that can self-replicate within a host cell. For example, plasmid. So then the second step is the generation of recombinant DNA. The gene of interest, that is the insert, is joined with the DNA fragment of the carrier DNA molecule, that is, if we take the example plasmid. So the plasmid DNA and the gene of interest are joined together. So that is known as the rDNA. The number 2 step is the generation of recombinant DNA. Number 3, that recombinant DNA is now transferred into a competent host cell. Host cell can be a microorganism, can be a plant cell, or can be an animal cell. Typically, we take in the RDT E. coli host cells. So number 3rd step would be to transfer these rDNA molecules into a host cell. Then number 4th step is the selection of those host cells which carry the rDNA and then allowing them to multiply, thereby multiplying the rDNA molecules. After selection of those cells which contain the rDNA, that host cells are allowed to multiply in a medium provided so that the rDNA molecules which have been inserted in them, they could self-replicate and multiply in numbers. So basically these basic steps are involved in a typical rDNA experiment. So this can also be understood by the help of diagram. This is the schematic representation of the basic steps in a rDNA experiment. So you can see the vector DNA and the DNA in which our gene of interest is situated. So both these DNA are cut with the help of restriction enzyme. The same restriction enzyme is used for cutting the vector DNA and the DNA fragment which contain the gene of interest. After cutting it with restriction enzyme, the two DNA, that is the vector DNA and the target gene containing DNA, that both are ligated. The vector which is shown in the middle with the blue fragment, it is the gene of interest and this vector is only to be cloned further. The rest two are of no use. So our recombinant vector is the one which is containing the blue fragment. Now this vector is selected and it is now inserted into the host cell. So this by the process of transformation. After inserting the rDNA, these cells are allowed to amplify in a nutrient medium so that the cells, the bacterial cells, divide and also the recombinant DNA which is inside that host cell, that also self-replicate and increase in number. Now we are coming to the facts and figures. The first recombinant DNA molecule was produced in 1972 by Stanford researcher Paul Berg. Berg joined together DNA fragments from two different viruses with the help of particular enzymes, restriction enzymes and ligase. Restriction enzymes such as EcoR1 are like molecular scissors that cut DNA at specific sequences. As the scissors cut a long ribbon into different sizes of fragments, the RE can cut DNA into small pieces at specific sites. And if the DNA from the different sources is cut with the same restriction enzyme, the cut ends can be joined together and then sealed into a continuous DNA strand by the enzyme ligase. So the two important enzymes are there, one for cutting and one for joining. For cutting, restriction enzymes are there, and for joining, ligase enzyme is there. After the production of first rDNA molecule, the first organism to contain recombinant DNA was engineered by Herb Boyer from University of California, San Francisco, and Stanley Cohen from Stanford University. Together, they introduced an antibiotic resistance gene into E. coli bacteria. We will see later that what does this antibiotic resistance gene does and why it is important in RDT. Now, Paul Berg was awarded the 1980 Nobel Prize for Chemistry for his fundamental studies of the biochemistry of nucleic acids, with particular regard to recombinant DNA. So these all are some of the facts and figures which you should remember for further use. So this is the end of part 1 of this chapter. I will continue with the next parts in upcoming videos. So please subscribe and press the bell button to get notification. Thank you.